High-fat diets are often blamed for weight gain, heart disease, or diabetes, but their role in autoimmunity is less publicly known and far more insidious. Beneath the surface, every indulgence in saturated fats and ultra-processed junk food may be feeding not just your cravings, but the smoldering fire of immune dysfunction. For individuals genetically predisposed to autoimmune conditions like lupus, rheumatoid arthritis, or multiple sclerosis, the damage from a high-fat diet isn't merely metabolic, it's immunological. The gut becomes the flashpoint, and the breakdown of tolerance begins. At the center of this process is the gut-immune axis, a highly regulated interface between the microbial world inside the intestines and the body's immune surveillance system. Under normal circumstances, this relationship is one of mutual restraint. Immune cells tolerate harmless food antigens and commensal bacteria, while the gut lining prevents inappropriate immune exposure through a tightly sealed epithelial barrier and mucus layer. Key players like innate lymphoid cells, ILC3S, secrete cytokines such as IL-22, which maintain this peaceful state by enhancing mucus production, antimicrobial defenses, and epithelial repair. But when a high-fat diet, particularly one rich in saturated fats and devoid of fiber, becomes a regular feature, that balance starts to fall apart. Dietary lipids alter microbial composition, reduce beneficial strains, and favor bile-tolerant and pro-inflammatory microbes like Bilophila wadsworthia or Escherichia coli. At the same time, the production of short-chain fatty acids like butyrate, which normally dampen immune overactivation and support regulatory T cells, begins to decline. What follows is gut barrier dysfunction. Tight junctions loosen, mucus thins, and microbial fragments such as lipopolysaccharides, LPS, begin to leak into systemic circulation. These fragments are not benign. They activate toll-like receptors, TLRs, on immune cells, triggering a cascade of cytokine release, IL-6, TNF-A, IL-1S set, that leads to chronic low-grade inflammation. But in people with autoimmune tendencies, this is more than just background noise. The immune system, already genetically primed to mistake self for non-self, begins to misfire. Chronic gut-derived inflammation trains antigen-presenting cells to become hyperreactive. Loss of oral tolerance. The immune system's normal indifference to dietary antigens occurs as food particles and harmless bacterial molecules repeatedly breach the compromised gut lining. These exposures increase the likelihood of molecular mimicry, where the immune system begins targeting host tissues that resemble bacterial antigens. In diseases like lupus, where antinuclear antibodies attack DNA and other intracellular structures, or rheumatoid arthritis, where joints are destroyed by immune complexes, such breakdowns in tolerance are central. Moreover, the gut acts as a training ground for regulatory T cells, TREGs, the peacekeepers of the immune system. But high-fat diets decrease the production of anti-inflammatory metabolites and alter microbial cues that promote TREG development. In their absence, Th17 cells, a more inflammatory subset, begin to dominate, producing IL-17 and further eroding immune restraint. This skewed balance has been implicated in the progression of many autoimmune diseases, including inflammatory bowel disease, psoriasis, and systemic sclerosis. And it gets worse. This immune shift is not confined to the gut. The cytokines and reactive immune cells spill into the bloodstream, cross the blood-brain barrier, infiltrate synovial joints, and travel to distant tissues. Once sensitized, the immune system doesn't need the gut's permission anymore. It begins attacking wherever the genetic or environmental terrain allows. In lupus, this may manifest as kidney damage or brain fog. In rheumatoid arthritis, as joint destruction. And all of it may be rooted, in part, in the slow, dietary erosion of mucosal tolerance. Alarmingly, this process doesn't require decades to unfold. Experimental studies show that short-term high-fat feeding, even within weeks, can cause enough gut dysbiosis and immune perturbation to initiate autoimmune features in genetically susceptible animals. In humans, we may not notice until the symptoms are systemic: joint pain, fatigue, unexplained rashes, or brain fog. By then, the gut damage is often deep, chronic, and poorly responsive to medication alone. There is also evidence that flare-ups in established autoimmune disease may be linked to dietary patterns. Periods of high-fat intake, especially in the context of low fiber and microbial imbalance, can provoke renewed immune activation. It's not just that the gut helps initiate autoimmunity, it may actively sustain or amplify it. In the end, a high-fat diet doesn't just feed the body, it trains the immune system to behave badly. And in people with the wrong genetic lottery ticket, that miseducation can mean the difference between a stable immune system and one that turns inward. Autoimmune disease, often considered mysterious or idiopathic, may in many cases have roots that are dietary, microbial, and disturbingly preventable. When the gut barrier falls, so too can immune tolerance, and the results are far from silent.